Now I'd like to introduce today's speaker. Felicity Keiper is an expert in global regulatory affairs in the seeds and traits business of BASF. She has worked in agricultural biotechnology for more than 20 years, with 14 years in regulatory affairs in the private sector, and prior to that, she was a research scientist in the public sector. She has a Ph.D. in agricultural biotechnology and is a lawyer. She works predominantly in regulatory policy with a focus on developments in international law that impact biotech R&D activities. Key areas include regulation of biotechnology, including synthetic biology, and of genetic resources in the context of access and benefit sharing and digital sequence information. In this talk, Felicity will give an overview of how synthetic biology and gene drives became topics of debate under the Convention on Biological Diversity and its protocols. Understanding the evolution of this topic within the CBD will give us much insight into how issues like this are raised and dealt with under the processes and procedures that Pete Vandermeer described to us last week. So it's my pleasure to turn over this session now to Felicity.
So as Hector mentioned, my work is mostly in the area of biotech regulatory policy, and my involvement in the work under the Convention on Biological Diversity and its protocols on the topic of synthetic biology, but also on biotechnology more broadly, has a regulatory focus. So I will be talking about regulatory related developments under these treaties in this webinar. And while I am from the private sector and I have that perspective, I have also been a public sector scientist. And so what I will be talking about does have that broader relevance to all members of the regulated community, whether or not they're public or private. So in this, in the first webinar of this series last week, Pete Vandermeer gave an introduction to the convention and the protocol, and also how they are implemented at national levels. And I am going to assume some of that knowledge and build upon on that in this webinar. And I'll only be giving a brief overview of the convention and the protocol. And then I'm going to spend some time talking about synthetic biology. This is a topic of quite a contentious debate under the convention, and gene drives is one of several subtopics within that debate that is receiving a lot of attention. So we'll go through the decisions that have been made on synthetic biology and also on gene drives to demonstrate how these topics have evolved over recent years, and then talk about the current status and what we can expect in the near future. So why do we have this focus on the Convention on Biological Diversity? It is the primary international forum discussing the regulation of synthetic biology. It's not the only international forum discussing synthetic biology, but it is currently the forum with potential regulatory implications for releases of GMOs, genetically modified organisms, into the environment. And the convention itself it's a broad environmental treaty, and it includes several provisions on biotechnology. Pete went through some of these last week, and I will talk about some of these in detail in a moment. The convention has been in force for almost 3 decades. This means that if it's in force, it means it's legally binding on its parties, on the countries that are members of it, that have signed up to it. And it's from that same era as other high-profile conventions like the Climate Change Convention. And it's one of the most ratified of the UN treaties, and it's considered near universal. The only UN member state that's not a party is the USA. So it has 3 main objectives, and you're going to hear me repeat the first 2 many times in this webinar: the conservation of biological diversity and the sustainable use of its components. And it has a 3rd objective of the fair and equitable sharing of the benefits arising out of the utilisation of genetic resources. Some more background here on this slide. The convention has subsidiary agreements, so there's a treaty family. And I will be talking about the Cartagena protocol in some detail, but not the other 2 that are shown here. This is for your information. So the Cartagena protocol has a supplementary protocol on liability and redress, and the Nagoya protocol sets out an access and benefit sharing regime. And that is a way of implementing that 3rd objective of the convention. So like the convention, the Cartagena Protocol has been in force for some time, almost 20 years, and it has a large membership, many parties. So last week, Pete mentioned several articles that were relevant to biotechnology, and I'm focusing here on two, and also the definition of biotechnology. And with Article 8G, I've showing it in detail here because I will mention it again through this presentation. So this is the obligation to regulate what it calls living modified organisms, which is abbreviated as LMOs. In practice, this means the same thing as GMO or genetically modified organism, and I will use both terms interchangeably. So this article requires parties to the convention to establish or maintain means to regulate, manage, or control the risks associated with the use and release of LMOs resulting from biotechnology, which are likely to have adverse environmental impacts. And those impacts are in the context of the conservation and sustainable use of biodiversity. So those first two objectives of the convention. So in essence, what needs to be regulated is the risk of a likely adverse environmental impact that is relevant to those two objectives. The convention provides a broad definition of biotechnology, which is any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. So this is incredibly broad, and I am going to talk about the definition of synthetic biology soon, so just bear this one in mind. And in Article 19, the convention includes an instruction for its parties to consider the need for a protocol setting out regulatory procedures. And this was the basis for the development of the Cartagena Protocol, which entered into force 10 years after the convention. So the protocol provides a regulatory framework covering the transboundary movement, transit, handling, and use of LMOs that may have adverse effects on the conservation and sustainable use of biodiversity. So this language, it ties it back to the convention and those two objectives. The protocol is often referred to as being concerned with transboundary movements, which it is, but that is not at all. The word use is very broad, and its procedures or procedures consistent with it can also be applied to releases of GMOs within national borders. Now, the protocol has several features. Peg talked about some of these last week, and I'm focusing on those here with particular relevance to the discussions on synthetic biology and gene drives. The definition of an LMO is in the protocol. So the convention uses the term, but it doesn't define it. The definition is found in the protocol. And LMOs have a novel combination of genetic material obtained through the use of modern biotechnology. This is an abbreviated definition here to simplify it a bit for presenting. But it defines the term, also defines the term modern biotechnology. And this is broad also, but it's more specific to biotechnology, biotechnological techniques than the definition of biotechnology in the convention. The protocol also enshrines something called the precautionary approach. And this is included in this is included in the objective to the protocol in its Article 1, and it's also referred to in the convention. And where this originates from is the Rio Declaration of 1992. So this is that era where there was a lot of soft law developed around environmental law. So the convention states that in regard to precautionary approach, where there is a threat of significant reduction or loss of biodiversity, lack of full scientific certainty should not be used as a reason for postponing measures to avoid or minimize such a threat. So in other words, this approach can be used to justify decisions that prevent the release of an LMO. So this can be on the basis of the decision maker not being satisfied that there is conclusive evidence of no risk or conclusive evidence of a level of risk that's acceptable. And this is quite a controversial, sometimes it's called a principle, it's called an approach, because it can be used to justify obstructive actions in a sort of a non-transparent way or an arbitrary way, like a ban or a moratorium on technological development or environmental releases. And at the centre, well, at least in my view, it's the centrepiece of the protocol, because I'm dealing with risk assessment requirements in my day job, the risk assessment framework. And this is used for the identification and evaluation of the likelihood of those adverse effects that are referred to repeatedly, the likelihood of adverse effects of LMOs on the conservation and sustainable use of biological diversity. So the risk assessment is the major source of regulatory requirements. So the information that regulatory agencies ask for is mostly for informing the risk assessment, and what is required for risk assessment is a major area of ongoing discussion and work under the protocol. So the protocol sets out several principles of risk assessment, and these remain just as applicable to the LMOs of today as they did 20 years ago. Risk assessment should be carried out on a scientifically sound and transparent, in a scientifically sound, transparent manner. It should be done on a case-by-case basis, depending on the LMO, how it's going to be used, so the intended use, and where it's going to be released, so the likely receiving environment. So this means that the information that might be needed to inform a risk assessment, it will vary case by case. There's also the comparative approach. This means that the potential risks of the LMO, they're assessed in comparison to the risks presented by the non-modified or parental organism. The protocol also provides for risk assessment, risk management measures. So these can be imposed if these are necessary to prevent adverse risks, adverse effects that may be identified in a risk assessment. So these are meant to be imposed to the extent necessary, meaning that they should be proportionate. So I've covered all of that background because it is relevant to today's discussions on synthetic biology and gene drives under the convention and the protocol. So now I'm going to switch to developments on synthetic biology and where discussions began on this under the convention. And they began in 2010. Biotechnology and LMOs have been discussed for a much longer time under the convention, but there was a shift in the terminology used from around 2010. And the convention started to sort of grow from this mechanism that the convention has, where it's parties as well as governments who are not parties, so that's the USA, and relevant stakeholders can submit proposals on new and emerging issues that are relevant to the conservation and sustainable use of biological diversity. So generally speaking, this mechanism was used to propose that synthetic biology is a new and emerging issue. And some of those proposals called for a moratorium. This was on the basis of the precautionary approach. Concerns were raised around uncertainties regarding potential adverse impacts. And so there were calls for the science to be stopped until regulatory rules could be assessed for adequacy and any gaps in those filled. So a trigger for this was likely included publications like the one shown on the slide, so where researchers reported the first cell controlled by a genome that was computer designed and chemically synthesized. So at the time, there was a lot of media attention about this and the discussion, and a lot of discussion about what might now be technically possible. So this new and emerging issues topic remains a contentious one today for synthetic biology. There is a formal process for identifying these issues that involves the consideration of evidence against a set of criteria, And those criteria have been defined by the parties to the convention. They're set out in a decision of theirs. So this consideration is carried out by the convention's subsidiary body on scientific, technical and technological advice, or SBTA. So this body meets regularly and it considers these proposals. And this is like one item on its agenda. The SBTA then makes recommendations. So they produce a document with a set of recommendations, and then that's the basis for parties to make decisions on the necessary actions. So the new and emerging issue assessment has not been completed for synthetic biology. So an early attempt was made by SBTA, but this was inconclusive. They didn't have enough information in order to be able to form a conclusion. And it subsequently made recommendations, this is SBTA, for programs of work examining many synthetic biology topics. So some of these are connected to the new and emerging issue criteria, but that sort of full assessment has not been directly addressed by SBTA again. The programs of work have included the establishment of ad hoc technical expert groups, or RTEGs, and an RTEG has, two RTEGs have talked about these criteria. One of them considered them in detail, that was in 2019. And there were participants in that group that considered some of the criteria to be relevant to organisms containing engineered gene drives. At the bottom of the slide there are references to the relevant documents. So what is a program of work? I'm going to say this term a lot too. Since 2010, the convention's parties have made decisions at 5 COPs on synthetic biology, and I am going to go through those decisions in a moment. So a COP is a conference of the parties, and prior to the COVID-19 pandemic, COPs met every 2 years. And this is a meeting of the parties to the convention, and it's the treaty governing body. It makes decisions, it reviews and makes decisions about implementation of the convention's provisions. And those decisions can include establishing work programs to examine specific topics in more detail. And the decisions relevant to synthetic biology over the years have established several programs of work, and these have examined many questions. For example, what synthetic biology is, so how to define it, what it includes, if synthetic biology qualifies as a new and emerging issue, as I've just talked about, what the potential positive and negative impacts may be regarding the conservation and sustainable use of biodiversity, what applicable regulatory framework or frameworks or mechanisms already exist, and are there gaps that need to be addressed, if the organisms created using synthetic biology techniques are LMOs as defined by the Cartagena protocol, and more recently, efforts have been focused on establishing a process for monitoring and assessing developments in synthetic biology so that issues can be identified. And you're probably hoping that at some point in this presentation, I was going to give you a definition of synthetic biology, but there is not actually one that has been agreed, at least in this forum. In my view, that very broad definition of biotechnology in the convention remains applicable. An ad hoc technical expert group attempted to do this. They were tasked with developing an operational definition, and they proposed one to the parties at a COP, but the COP didn't endorse it or officially adopt it. So sometimes it's presented as though it's an official definition, and it is used in discussions under the convention, but it's not an official definition. There's also no agreement on what synthetic biology includes. In the discussions under the convention, there's this sort of ever-expanding list of enabling tools, technologies, applications that are labelled as synthetic biology. And what the term may mean to a researcher or to a technology developer can be very different to what it could mean in the discussions under the convention. There are many different views on how this should be defined and what it includes. And there's no clear scientific boundaries. The way that I see it, which is just my own personal view, is that the boundary in these discussions is the entry into force of the Cartagena Protocol. So the boundary is a time rather than a scientific development. So everything that has emerged since then is being sort of listed in what's considered and being debated as to whether or not it remains within its regulatory scope. And it's labelled as synthetic biology, because there's an argument there that when the Cartagena Protocol was drafted, it didn't foresee developments that would come afterwards. So what has been agreed in all these almost or more than a decade of work programs? We can't define synthetic biology in this forum, but we can agree that it falls within biotechnology as defined in the convention. So this means the regulatory obligation of Article 8G applies. Another major outcome is that there is agreement that most organisms developed using synthetic biology techniques are LMOs as defined by the Cartagena Protocol. So this means that they are within its scope and subject to its regulatory obligations. So they remain regulated, and this includes organisms containing engineered gene drives. So I have quite some detailed slides coming up that go through the main points in the COP decisions relevant to synthetic biology and gene drives. And these decisions, especially the more recent ones, are quite lengthy documents. And for each of them, I've pulled out the indicators of where the discussion is heading. And over time in these decisions, you can see incremental changes and issues kind of building and expanding. So beginning with COP10, 11 and 12, these decisions were made under the COP agenda item of new and emerging issues. So the document and decision numbers are shown underneath the dot points here. And there was also mention of synthetic biology in a decision on biofuels back in 2010. And a constant in these is reference to the precautionary approach. This is in regard to environmental releases. And in 2010 and 2012, there were calls for submission of information addressing the new and emerging issue criteria. And this information was to inform that assessment by Substar according to the prescribed process that I spoke about earlier. In 2014, the COP decision concluded that there was insufficient evidence to complete that new and emerging issue assessment. And the program of work of synthetic biology was significantly expanded from that point. This included the first of 3 iterations of an ad hoc technical expert group. This was established and it was mandated to consider a list of topics, and these included, among other things, developing a definition that I just mentioned, also examining the scope of existing regulatory mechanisms and existing risk assessment practices. And the work of the RTEG was to be informed by submissions of information and a series of online discussions on the same list of topics. So here in 2014, there was a shift from information being submitted for consideration by Substar to an RTEG. So an RTEG is a much smaller group. Substar is like a mini COP that includes delegations representing all of the parties. The RTEG has maybe 30 parties represented and a small group of non-parties or observers. And what the RTEG does, it's tasked with considering a greatly expanded volume of information. With these online discussions and information submissions, there are hundreds of pages of information. And they distill that down into a series of recommendations for Substar, who then in turn make recommendations for the parties to make decisions on at COP. So to explain some of this a little bit further, where information is invited, this is open to parties, but it's also open to other governments and to relevant stakeholders. And that is in the form of a document that is submitted to the Convention Secretariat. So quite detailed responses to the questions, so the topics can be submitted. Registrations to participate in the online discussions are also open to these groups. Participants of the RTEG, while they are predominantly parties, other governments and relevant stakeholders can also be nominated. And there's like a selection process, and they may be selected. So relevant stakeholders can include many groups, such as the private sector, and there are business groups that participate, and that's how I participate. But there's also many non-government organisations, NGOs. There are several organisations representing Indigenous peoples, and there are also representatives of the research community. So these work programs, there are ways of contributing to the discussions quite directly, even if you're not a party. In 2016, synthetic biology became its own standalone COP agenda item. So this is a move from being a few paragraphs within a bigger decision on new and emerging issues, which might include other proposals for new and emerging issues, to quite a lengthy decision just on synthetic biology. And here the precautionary approach is mentioned again, and this time its applicability to some LMOs containing gene drives is also mentioned. A definition for synthetic biology, that was the definition that was proposed by the RTEG, was acknowledged. This is acknowledged as a useful starting point for discussions under the convention, but not formally adopted. And as I've mentioned, there's a range of views on this. There are parties that are not happy with it and often raise reopening this and trying to develop something better. It was also noted here that risk assessment methodologies may need to be updated or adapted, and this is for organisms developed using synthetic biology techniques. The RTEG was continued with a new mandate and a new list of topics to consider. And also in this decision and the decision that followed at COP14 in 2018, which is on the next slide, the need for completion of the new and emerging issue assessment was recognised. And so there was a building pressure here from some parties to get this done. And the RTEGs were tasked with considering the criteria and providing recommendations to Substa. The RTEG mandate in this decision, while it was there, they didn't end up addressing these. They deferred the assessment, but the RTEG that followed, they did finally go through those criteria. So in 2018, and so this is the last time that COP has considered synthetic biology. You might notice some duplication in the topics assigned to the RTEG. The same topics were included for the invitations for submissions of information. It was getting a little bit repetitive for those of us participating. But the differences here, they're on the agreement on the need to establish a process for the broad and regular horizon scanning, monitoring and assessment of technological developments. Also, there are several paragraphs dedicated to gene drives. So here the issue is building. And one of these recognises that specific guidance may be useful to support the case-by-case risk assessment of organisms containing engineered gene drives. So this is additional guidance to what may already exist. Also, it was noted that the free, prior and informed consent of Indigenous peoples and local communities may be warranted if they may be affected by releases of organisms containing engineered gene drives into the environment. There's also mention of the precautionary approach again, but this time it is in the specific context of releases of organisms containing engineered gene drives into the environment. with these requiring scientifically sound risk assessment and risk management measures. Now, in parallel to the COP14 decision I've just talked about in 2018, and at the same biodiversity conference, there was also a decision of COP-MOP 9 to the Cartagena protocol. So the COP-MOP, which is also called MOP, this is the governing body for the protocol. So the protocols have their own governing bodies comprising their parties. And risk assessment and risk management are agenda items for COP-MOP. At every COP-MOP, there's usually some sort of work program on these topics. And the decision here in 2018 had some duplication with the COP14 decision on synthetic biology. So there was some agreement between them, especially on specific guidance, supporting case-by-case risk assessment, and the free, prior, informed consent of affected Indigenous peoples and local communities. That's what IPLC stands for. So the key points are that it established a process for identifying and prioritising specific issues that may require the development of additional risk assessment guidance. And a work program was established to apply this process to certain organisms, including organisms containing engineered gene drives. So in the work program, a study was commissioned. So this is contracted out by the Secretariat of the Convention. And in that study, that process was applied, and a new RTEG was established to review this work and make a recommendation. And this RTEG, this is a separate RTEG to the synthetic biology RTEG. There's multiple RTEGs with different mandates. And like the synthetic biology work, the outcomes of the work on risk assessment are also considered by SBSTA. So this is referred to the SBSTA and on their agenda. So this was the last time that the COP-MOP considered risk assessment. So this is where we are up to in terms of decisions today. Now, I've included this slide to provide some context for the environment in which the decisions were negotiated at the last two COPs. So the COP in 2016 and 2018, and also the COP-MOP I just spoke about in 2018. Here there were campaigns for global moratoriums on the development of organisms containing engineered gene drives and also environmental releases of them. The basis for these campaigns was that precautionary approach necessitated such a measure. And this could have been a decision under the convention, but such a drastic action was ultimately rejected by the parties. And instead we have these ongoing work programs. Synthetic biology is now a fixture on the convention's agenda. Developments in the field will continually be monitored. And we have seen increasingly prescriptive recommendations for work with gene drives, and there will most likely be the development of this additional risk assessment guidance materials. And these kinds of outcomes are avoided when parties who are making the decisions are well informed. You know, most of them are not going to have a gene drive expert sitting in their delegations in these meetings. So the engagement of the research community in the work programs that I've been talking about, but also the meetings themselves is important. And this occurred quite effectively with these COPs, where the research community reached out to parties to make sure that their perspectives were also taken into account in these discussions. And for example, there were open letters issued shortly before each of these COP meetings. So everything I've just talked about is publicly available. The convention has an extensive website. This has all of the COP decisions. It also has all of the work of the SBSTA. It has all of the submissions for new and emerging issues. And there is a synthetic biology portal. The Cartagena protocol also has a website that has all of the COP-MOP decisions, and it has a risk assessment and risk management portal. So in those portals, you can find the work programs that I've just been talking about, and all of the information submissions that have been made over the years, the online discussions, so the topics discussed, the posts made, and the RTEG reports. So it's all there. These slide has screenshots of the menus that are in those two portals. So taking a pause here again to change gear, I have talked about what has happened and where we're up to, but I'm now going to talk about what comes next. And I hope you're all still with me. I know this is a lot of complicated information. So I mentioned that the last COP and the COP-MOP decisions were in 2018. This was the last time that a full biodiversity conference was held. The next one was supposed to be in October 2020, and that logo is on the left side of the slide showing a 2020 conference, COP15, MOP10 for the Cartagena protocol, and also MOP4 for the Nagoya protocol. These are held in concurrent meetings over like a 2.5 week period. But all of this was delayed due to the COVID-19 pandemic. So prior to that, in the middle of 2020, there was supposed to be preparatory meetings, which included a meeting of SBSTA. There was also supposed to be another meeting that is developing, meeting of a body, a working group that is developing the post-2020 global biodiversity framework. And I will come back to that topic. But first, the SBSTA meeting needed to occur because it needed to review the outcomes of the programs of work mandated in the COP14 decision for synthetic biology and the COP-MOP9 decision for risk assessment. It is then supposed to provide recommendations for decisions to be made at COP15 and COP-MOP10. That SBSTA meeting was held in part last year over several weeks of virtual meetings, many weeks of virtual meetings. And then it was completed in mostly in-person meetings in Geneva in March just this year, so very recently. There will be, so those meetings are completed and the outcomes of that will go to COP15 and COP-MOP10. The working group developing the post-2020 global biodiversity framework is still developing that text, still negotiating aspects of that, and they will be having an additional meeting in the middle of this year. So the Biodiversity Conference was held in part in October last year to address some of the administrative items on the COP15 and COP-MOP10 agenda, also the COP-MOP4 for Nagoya Protocol, but it didn't go into substantive agenda items like synthetic biology and risk assessment. Decisions, negotiations and decisions on these items will be on the agenda of part 2 of the Biodiversity Conference. And this is expected to be held in person, still in Kunming, China, in the third quarter of this year. At COP15, we can predict what will be negotiated for synthetic biology based on the recommendation from the SBSTA 24 meeting that's just concluded. So that recommendation recognises that the new and emerging issues analysis has been inconclusive for synthetic biology, and there is bracketed text around options on what should happen next. So if the text is bracketed, that means it has not been agreed. It's likely to be a contentious area. And the bracketed options will need to be negotiated and resolved if they're going to be at COP15, if they're going to be included in the eventual COP15 decision. It also sets out a process for the horizon scanning, regular horizon scanning, monitoring and assessment. And much of that detail is also bracketed, such as the body who will carry out the work, how long it will operate for, and who is tasked with what in the various steps in that process. So, and quite a lot of that text was not actually considered at SBSTA. So while there's still a lot to be resolved here, the previous COP decision is to establish this process for horizon scanning. So it's a pretty safe bet that this will be an ongoing area of work in the near future. And this is a change from previous years where the work program did include a form of monitoring developments. This was part of the information submissions and the online discussions, that the topics that were discussed there. But those work programs had several additional regulatory related topics. But the process does include this assessment step, and it's not clear at this point what that will involve and what the information from that will be used for. So it's important that the scientific community is engaged here and it is working, those that are working in the field are contributing to and informing the whole process. At COP-MOP10, we can predict what will be negotiated based on, for risk assessment, based on the recommendation also from the Subsidiary 24 meeting. And that recommendation endorses an outcome of the work of the RTEG, which was to recommend the development of additional guidance materials to support the case-by-case risk assessment of LMOs containing engineered gene drives. And it recommends establishing a new RTEG to do this work, with their work to be supported by input coming through information submissions and online discussions. So this looks more like a typical program of work where there's a range of opportunities for participation. But there is still, in this text as well, there is bracketed text, unresolved text around what the RTEG should be doing. So the drafting process for the guidance and the focus of the guidance. For example, should this be mosquitoes or should it be broader? So all of this text needs to be negotiated at COP-MOP10 and resolved for the eventual decision. Now, something that I've only mentioned in passing so far is the post-2020 Global Biodiversity Framework. And I'm not going to go into detail on this because it is a very large topic on its own. But I just wanted to flag that this is a 10-year or possibly even longer plan for the convention and its protocols. And it contains a target on biotechnology. And the text in this target is still a draft under negotiation, is currently heavily bracketed with many text options. So at this point, it's not easy to predict what it may eventually look like. And it will be negotiated, or it may be negotiated at that upcoming meeting of the working group in June, or it may go straight to COP15 for negotiation and resolution, along with resolution of the broader framework and its adoption is supposed to take place at COP15. So the basis for this target is Article 8G of the Convention, and this has the obligation to regulate. And that's the article that I showed you earlier in this presentation. And the content of this target, it's relevant to what we're talking about because the scope of it could potentially be broader than Article 8G, and it could be the basis for future regulatory discussions, programs of work, and developments under the Convention that impact biotechnology and everything that's labelled synthetic biology. So it's something to be aware of and watch how that develops. And this is my last slide. Hopefully I'm doing okay for time. And this is just to briefly mention another international organisation, because this has a mandate that is directly relevant to the Convention. This is the International Union for the Conservation of Nature. So it's concerned with conservation, and that is one of the major objectives of the Convention on Biological Diversity. So the IUCN is a large group with representatives of governments, of NGOs, and also the academic community. Those of you watching this right now are probably much more familiar with this forum than I am. It's not something that I've actively participated in. But the main point here is that the outcomes of IUCN meetings and work programs can influence what CBD discussions and developments there. And they have a program of work on synthetic biology in parallel to the work programs under the Convention. And an outcome of that so far is a report that is an assessment of synthetic biology and biodiversity conservation, and gene drives are in the scope of this work. And at their most recent Congress, which was delayed from 2020 and held in September last year, there was a resolution to establish a program of work for developing a policy And this is on the implications of the use of synthetic biology in nature conservation. So there's a Congress every 4 years. It usually is held a few months just before every second COP. The next one, it will be in 2024, and the outcomes of that program on the development of a policy will be deliberated. So there could be a policy, or there could be a continuation of the work to develop this policy. So with that, I will conclude the presentations. And so thank you for your attention. I hope you're all still with me. Happy to take any questions, Hector, if there are any.
Yes, yeah, there are. Thanks, Felicity, for that very detailed and thorough discussion on how this CBD came to deal with synthetic biology and then gene drives. We have a couple of questions already on the screen here, and one person who has raised their hand to ask a question. But before we do that, I'd like to begin by asking my question, which has to do with the operational definition of synthetic biology. It seems like it when the CBD has talked about operational definition, it seemed to me at the beginning that it was just a temporary definition that it would allow for further work, but it's already been the basis for a lot of effort and document writing. So wouldn't that in practice mean that the operational definition becomes an official definition de facto? And doesn't the fact that products of synthetic biology so far have been classified as LMOs, doesn't that mean that it becomes part of the protocol? And I guess the question is whether the synthetic biology definition, even though it has been an operational / in my view, working definition, that it might already be irrelevant to the actual, whether that it actually impacts the work of the CBD and the Cartagena protocol, since work has already begun and will continue to proceed.
Yeah, so the operational definition, I agree that in practice it has kind of become a de facto definition. It is cited. I just saw part of the launch for the synthetic biology, new synthetic biology technical series, which is based on this definition. There wasn't a lot. I participated in the RTEG that developed this, and it was very difficult discussion because there are just very different views on what it is. There is the view that's my view, that it's not really necessary to define it, because biotechnology, as already defined, is broad, and it continues to capture the continuum of biotechnological development. But there's also the views that whole list that I showed on a slide, and more than what's on that slide, is synthetic biology. And this is all something new and different. So this is difficult to reconcile. And there are parties that are not satisfied with the definition because it's not specific enough. It makes it very difficult to address things like the new and emerging issues analysis, because it's not really clear what you're applying to the analysis. But in terms of regulatory scope and what's an LMO and what's modern biotechnology, my personal view is I don't think it's actually important to define synthetic biology, because what's more important is that the definitions that already exist continue to apply, and that means the regulatory mechanisms that already exist continue to apply.
Yeah, that kind of reflects the question that I asked at the very beginning several years ago, was just, is synthetic biology really a thing? And does it matter to actually define it? But I'd like to turn the mic over now to someone who's raised their hand, and I hope the technical staff can help me by unmuting his or her microphone. Dodu, are you still wanting to ask a question? Let me see. Can somebody help me by unmuting the microphone? Oh, okay. It looks like you're ready to talk. So go ahead, please. Hello? Are you still there? We can't hear you. We'll keep trying. In the meantime, then, I'd like to then ask Wayne Landis's question. Wayne asks, has there been any consideration of the detailed description of risk assessment framework and risk management considerations that have been found in the U.S. National Academy of Science, Engineering, and Medicine 2016 Gene Drives on the Horizon? There are concrete suggestions for how to conduct a risk assessment for a gene drive organism in that document that would be useful in forming a basic structure of a risk assessment.
Yeah, so in this program of work, this is part of what's contentious about what the RTEG will be doing. Where there is a decision to develop something like additional guidance materials, it's important that there is a process for this that enables a useful outcome. So there is this is the view that we've been contributing, is that process should involve an assessment of what already exists and where there are gaps that need to be filled. So what needs to be addressed in additional guidance materials that isn't already covered by existing guidance materials. So it's a bit of a contentious topic because developing guidance under the Cartagena Protocol. This is about implementing the Annex 3 in the Cartagena Protocol. There's a history here where guidance documents have been developed that have not ultimately been adopted by the parties because they haven't been useful, or they're not used for their intended purpose. They might be used for training, capacity building purposes, but there's a bit of a tension here because what already is developed isn't developed by parties to the protocol or parties to the convention. It may not have been brought to the attention of the work program here, or not adequately sort of amplified in that forum. So that's why we've taken the position there that the RTEG needs to be doing an assessment of all of these materials that already exist, because materials already exist, and adding something useful to that.
Yeah, so I guess that also brings to mind a question that relates to the development of risk assessment guidance that I have, which is whether, what impact this additional guidance that were it to be developed would actually have. There's some language that's attached to the charge for the development of this additional guidance that makes it seem as though it's an optional thing.
Yeah, so these guidance materials are supposed to be voluntary, but they can be authoritative, like they can be relied on by parties, particularly those who lack experience in conducting risk assessments, especially for risk assessments for releases into the environment. So these are materials that can contribute to training and capacity building. So they can have an impact. So that it is important that they are useful, in that they are practical and actually assist with conducting a scientifically sound risk assessment.
With regard to your comment about being useful, I also would like you to comment on the existing guidance that's been developed, and also the Annex 3 itself as a sort of an overarching framework for risk assessment, and how well that even up to now has been implemented and has been able to be internalized by people in different in different countries.
Yeah, so there is a history under the protocol with many programs of work. I think the first one on risk assessment guidance, it might have started around 2008. And at every COP-MOP, so every 2 years, there's like a continuation of or renewal of these work programs. And there was a risk assessment guidance document, so a more general one to assist with the implementation of Annex 3 of the protocol, that was developed over a long period of time. It went through several rounds of revision, of substantial additions, of testing, and ultimately what was produced. So there's a document, you can find it on the convention or on the protocol website, but it wasn't officially adopted by the parties to the Cartagena protocol, not officially endorsed. So it's recognised that it exists, but it's not promoted as something that is an official document. And there is a disclaimer attached to it that doesn't reflect a consensus view of the RTEGs that worked on it. So it was quite a contentious series of programs of work. And what exists now is not well used for its intended purpose. In my own view, so I find the document incredibly complicated and difficult to follow. And I'm quite familiar with general requirements of risk assessment in several countries. So if someone was trying to use that to assist them to do a risk assessment who didn't have some of that background familiarity or prior experience, it would be very difficult for them to carry out a risk assessment. It's almost so complicated that they would never be able to complete one. And part of the problem there was the process for its development. And that's what's recognised in the discussions now around what the RTEG should be doing, who is mandated with developing additional risk assessment materials for gene drives. So there needs to be a process that will improve the approach taken and the likelihood of a useful outcome. So a lot of the input there is aiming to sort of focus that on a particular topic and the way that it's drafted. So it's the end product is something useful that adds to what already exists, if it is even necessary to add to what already exists.
Great. So yeah, I think it's good to note that this additional guidance would be on top of the main document, which already has the status which has the kind of status that you had mentioned. But, Dave Obrakta has his hand up, and so I'll turn the mic over to him.
Thank you, Hector. Thanks, Felicity, for a great talk. It was really informative, at least from my perspective. It was. There was a lot of gaps in my mind to fill, and you did fill them. I had a question. If you could, maybe it's a speculative question, and I wanted you to speculate sort of on a counterfactual here. That is, could you think about what if that moratorium had been the decision at COP was that to I don't know how you would frame it, but they would adopt the moratorium, and at say at in 2016 or whenever that came up, and could you sort of step us through, if that occurred, then what would happen next, and how things might look today in terms of how it might be impacting, say, researchers and research and development in gene drives?
In my view, I would be extremely surprised if something like that was successful, because it's such a drastic action. But an outcome could be, like we've got now, work programs, but something a bit more restrictive, like something that limits releases into the environment until we have, for example, a risk assessments guidance document or something like that. So it could, the impacts of that could be, delays in research programs and a lot of uncertainty about where that ends, and where activities might be able to be resumed and under what conditions, if that pause is on while new rules are being developed. So well, I guess anything's possible, but I would be surprised if there was such a moratorium. But things can happen that delay progress in research. But there's also the question, if there is a COP decision for a moratorium, is that actually binding? Is that something that's legally binding? Because it's the treaty that's legally binding on its parties. So a COP decision is not the same thing, and if it's not a treaty text, it's consensus text. Well, it's called consensus text, but sometimes what's in it may be something that's insisted on by quite a small number of parties. And in this case, a moratorium, that would have been the case, a very small number of parties supporting that. So, and whether or not something like that would be binding really goes back to the text of the treaty and the powers given to the COP to do these kinds of things. And there are, powers of the COP that are set out in the convention, and they're things like amending text to the protocol, amending text to the convention. So at worst, something like that could be introduced there. But that would require all the parties to agree to it. The parties that are, to the convention would have to agree to that change.
So if I could just jump in. So if the decision is made at COP, and again, I'd like to use the moratorium as an example, just to imagine what would a decision made then become, would the parties then immediately act on a decision that's made at COP? I mean, how does it filter down to the parties? Do they just say, okay, a decision has been made, and we, the party that I belong to is going to honor that decision by doing what? I mean, how do they honor that decision?
Well, that is possible. Even if the COP decision itself is not legally binding on all the parties, a party can decide to implement something in a COP decision in their national laws and regulations, if it decides to. So then it becomes legally binding within that party.
Okay. Yep. Thank you.
So as a kind of a follow-up question to that, you had mentioned that something like a moratorium could enter into a COP decision through what's called a consensus text, but I'm confused when the term consensus text is used, but as you say, it might be only supported by a few members. How does that work? How does something actually end up being a COP decision?
Yeah, so before, on these particular topics, they are, so all the outcomes of the work programs are considered by SBSTA, and then SBSTA develops like some preambular text and then some recommendations and some suggested text for a COP decision. So that becomes the basis for negotiations at COP. So it kind of gets 2 rounds of negotiations. It's the sort of more scientific and technical input goes in at Substa, and then it's negotiated. More of a political discussion goes on at COP. So there can be things that there are things that can be introduced to text in the very last minute. There can be things that are introduced quite early on and are insisted on very strongly by a small number of parties. And this, or a sort of maybe a watered down version of it, can end up in the actual decision document. If it's in the decision document, it doesn't necessarily mean it'll be acted upon, but it creates the potential that it can be.
What about the idea then that if in fact there is some guidance that's being worked on by a risk assessment ATAG, what potential is there for, say, a decision to be made that says, well, until this guidance is in place, there should be a halt to research on this topic?
Well, that is a possibility. Like in terms of the moratorium question that David raised earlier, there could be a moratorium or just restriction on activities until something like that is in place, until new rules are in place, guidance on risk assessment are in place, things that are considered to be gaps that need to be filled. So that's what the moratorium campaigns were about in the first place, that we need to stop the science until we fill all of these gaps. We need to assess what's needed to have adequate regulatory oversight. And until we have those things in place, we need to stop the science. So that's the basis of these moratorium campaigns. Whether or not they're successful, the very early submissions for synthetic biology in 2010 were very similarly worded. We need to have a global moratorium on everything. Over time, that has narrowed down to gene drives, because these are, I mentioned that these are identified in some of those criteria for new and emerging issues. And this is around, some participants in the RTEG considered that there was too many uncertainties. So when you have too many uncertainties as to potential adverse environmental impacts, that's when the precautionary principle kicks in. So that these kinds of actions are necessary to make sure that you can have adequate regulatory oversight to deal with all of these questions. So these things are not impossible, but I think what's more likely is that you have sort of these incremental steps where you're doing more work to try and uncover gaps, if they are even there, and what to address.
Great, thanks. Pete Vandermeer has written a long question, so rather than read it, I'm going to take this opportunity, if I can ask for his mic to be unmuted. Pete, I hope this doesn't put you on the spot, but I'd like you to ask your question yourself and maybe even initiate a discussion around the topic.
Good morning, Hector.
Hey, thanks for joining.
First of all, my compliments to Felicity, a very clear presentation, as we know, and that somewhere in the early hours of the morning, right? It must be 2 o'clock or something. I'm so impressed. Mine wasn't really a question. I just added 2 thoughts. One is on the and I don't know whether everyone can read it, so I can repeat it. One is on the definition of synthetic biology, because I think Hector hit the nail on the head. People think about it is a thing or a technique. No, it isn't. And I just quoted what an iGEM student said very wisely once, that SynBio is not a particular technique. It is a mindset of how you use a whole broad variety of techniques, ranging from molecular biology to artificial intelligence, and that all on the basis of design. And I think that is the key thing in there. But if you think about SynBio as a thing, and then you get frustrated that there is no definition, it becomes difficult. And then the other discussion about risk assessment. First of all, organisms developed through SynBio the risk assessment for that can be done without any problem on the basis of Annex 3. The methodology is there, it can work. And we should not mistake the idea of guidance. There is a gap. It doesn't mean there's a gap in risk assessment. No, guidance is there to conduct the methodology of Annex 3 in an efficient way within the timeframes. Guidance is not there because there is something wrong or missing in Annex 3. And that's often a misunderstanding. And if you think about guidance that way, then I can understand that people would say, oh, if there are things really missing in Annex 3 and we need guidance to fill that, then there should be a discussion and a moratorium. But that is not the basis for guidance. Guidance is additional material to make life easier. If there were things wrong in Annex 3 that you could not do a risk assessment, then the COP decisions or conclusions would say that, but that's not what we're talking about. If you look at the latest decisions of or draft decisions formulated by SBSTA, it talks about additional guidance materials to assist risk assessment. So there is nothing in there that would suggest that the current risk assessment methodology, the paradigm in Annex 3, would not be applicable or not be sufficient. Guidance is there to make your life easier, and in particular for countries that have limited resources. And I would add to that, is that also in particular for countries that wish to be more efficient in the way they use their resources. And it is something we need to underline in the negotiations. If we talk about gaps, it's not a gap in risk assessment, it is a gap in tools to make things easier. My apologies, because I know I spoke already enough last week, so I'll go on mute again. If I have no idea how to do that, but there we are.
Yeah. We'll take care of that for you, Pete. But thanks for your input. Felicity, do you have any comments or response to what Pete just said?
No, that I agree. That is quite an important point, that the guidance is to help with the implementation of Annex 3. And I think you asked that question earlier about my views on Annex 3. And I do have some criticisms of the guidance that's been developed before, but not of Annex 3. I mentioned the principles of that, and the methodologies remain sound today, irrespective of the type of LMO. And in particular, that case-by-case approach to risk assessment, that's very applicable, because as types of LMOs may change. And I think some of the concerns people have around risk assessment for gene drives is what kinds of questions should be asked, because they're different to the kinds of questions that need to be asked for, I work in crops. Crops you plant it stays where it is. So there's different sort of environmental, different questions that need to be asked for an environmental risk assessment. So the guidance, I think people are seeking that kind of guidance. What are the questions that need to be asked? And do we have that kind of information available?
So I guess this discussion so far, it really has something to say to those, such as the group I work with, who are concerned with capacity building, particularly in the area of risk assessment. And that is that most countries will be looking for, when we're looking at capacity building efforts, looking for guidance to help implement what is their obligation, really, as part of as parties, which is to implement risk assessment consistent with Annex 3. And that would be the most helpful form of capacity building that would be useful for parties that we hope to engage in any kind of capacity building effort. Dave has his hand raised again, so he has another question.
Yeah, thanks. Thanks, Hector. Thanks, Felicity, and thanks, Pete, for the information that you interjected there. I thought that was really helpful. Also, Felicity, are there certain parties that seem to have dominant roles in these proceedings? You mentioned that there were some parties who, felt like there needed to be more detail or something. And is there, based on your experiences, certain parties that play a dominant role in these discussions?
There are sort of groups of like-minded parties that, so there's a sort of I'd say it's a sort of a more precautionary block of countries or parties. And then there's at the other end of the spectrum, there's a number of parties that are more, I guess, more science-based or more innovation enabling. They want to sort of enable technology so that they are able to realize the benefits of it. So there are sort of these camps, and then parties that are somewhere in between. So yes, there are definitely on a lot of things, particularly on biotechnology, and for a long time, since the beginning, polarized views on how things should be regulated, the rules that are necessary, how precautionary things should be, whether the focus should be on regulating all of biotechnology, or if it should be a process-based or technology-based approach versus looking at the case, the case-by-case approach, looking at the characteristics of the LMO and the actual risks that present. So there are a number of different approaches. There's different ideologies. There's different, quite a bit of polarization in these discussions. So quite often you can end up with a document, and you see it with the RTEG reports. So that's very evident in the RTEG reports. And that was something that I participated in, where if you weren't part of that meeting and you read the report, it doesn't make a lot of sense. Like, you don't really get any messages out of it, apart from the big things like most organisms are LMOs as defined by the Cartagena protocol. And that's because there's such a polarization of views, and they're very difficult to reconcile. You end up with something that's in between that nobody's really happy with because it doesn't represent what they wanted to achieve.
Yeah, great, thanks.
That one comment that you made regarding reading the reports and being and whether or not they reflect what one might have taken home from the actual discussions was a question that I actually had when I was following the discussions around the risk assessment ATAG regarding whether or not there should be additional guidance. Of course, there were two questions, one being whether additional guidance should be developed for fish, LM fish, and then the other for gene drives. And it seemed to me that the discussion in both cases were pretty much similar, but the resulting decisions or the resulting conclusions were quite different. One being that there was no guidance needed for LM fish, but that there was for gene drives, and I couldn't, for the life of me, this is just me talking, figure out what tipped the balance between one decision and the other based on what I was able to see in the online discussions. But that may be something for another day to discuss. I don't see any more questions, either people raising their hands or entering them on the chat. And so I'm going to give you 10 minutes of your precious time back. Well, actually, it's more like 7 minutes of your precious time back, and hopefully that will be translated into sleep for you. So I really appreciate your willingness to give this presentation. It was a sacrifice that we asked you and required more of you than our normal speakers. But anyway, thanks very much.
You're welcome, and thank you for inviting me to participate.
Yeah, this is quite useful. And for those of you who, again, I want to make sure that you remember that this presentation will be posted online if you want to refer back to it as a resource on the GeneConvene website. So before we leave, however, I'm going to make sure that you are aware that next week we will be having a presentation by Dr. Musa Savadogo, who is with the African Union Development Agency, the new partnership for Africa's development. We're going to be entering a different phase of the webinar this time, where having these two presentations given by Pete last week and Felicity this week, talking about the CBD itself and the and the issues that have evolved that are relevant to those who are interested in gene drives. We're now going to give the opportunity for different stakeholder groups to talk about their particular take on these issues. So the first one is going to be from the African region, and Musa Sabadogo will be trying to outline to us the perspectives from the African continent on gene drives, synthetic biology, and what his organization is doing to support the African group of negotiators at the CBD negotiations. So with that, then I'm going to thank you for being online with us and hoping that you can join us again next week for our continuing series on the demystifying the CBD. I should also add that Musa will be making his presentation in French, but it will be translated into English. So I hope you can join us then, and thanks again.